Hey everyone, I'm David. I'm a medical science student at Western University. I just finished my second year, and here is a summer of my life doing research at Harvard Medical School. Alright, first day done. I am absolutely exhausted. We woke up at 7 a.m. this morning to drive from Ottawa to Toronto, which is about a 4-hour drive, and then I flew from Toronto to Newark and then Newark to Boston, which is about 5.5 hours in total. Finally arrived at my hotel at about 10 p.m. It is currently about 11 p.m. I'm probably gonna hit the gym just because it's the first time in months that gyms haven't been closed in my near vicinity, because back in Ottawa, due to the restrictions, all the gyms around here are closed, and at school at Western, all the gyms are closed as well. So finally we have an open gym. I'm gonna take full advantage of this. And it's probably a terrible decision because my legs are already cramping from walking around the airport so much today, but I'm gonna do it anyways. And tomorrow, hoping to sleep in a little bit, grab some breakfast at the hotel, and then check into my residence for the summer. Yeah, that's what I have planned, and we'll check in with you tomorrow.
Take your time, I'll be right here, I know.
Good morning. So it's about 8:30 in the morning. This hotel offers complimentary breakfast, so you know how to make the most out of this. And I got some breakfast and some lunch, pretty standard stuff, some bacon, bagels, pancakes, sausages, eggs, tater tots. But this food is actually really, really good. I usually don't like hotel food, but this is definitely 5-star breakfast food. Shout out Embassy Hotels. So right now, I'm gonna do a little bit of light reading. I have a couple research papers that were recommended to me by my supervisor, so I'm gonna tackle those, as well as learn a bit more about the people at my lab. What projects are they working on right now? What projects have they been working on recently? What are their backgrounds in terms of research interests? And yeah, hopefully that will prepare me a little better when I meet them tomorrow. So here I am in my room in artist residence. I literally haven't unpacked anything yet, but I just moved in and I'm super stoked to be living here this summer. First off, artist residence is one of the many residences offered by Massachusetts School of Arts and Design. Earlier this year, I was really struggling with looking for housing because I really wasn't sure, did I want a roommate? Did I want to live with a host family in Boston? Because of the pandemic, I was pretty worried about living with other people, so this was just a perfect option for me. I'm currently living in an apartment which has 4 different bedrooms, but since the other 3 bedrooms are unoccupied, I get all of the amenities to myself, so it's an absolute steal. I have a full-on kitchen, a lounge, bathroom, everything I need, and it's great. The location is also prime. It's about a 10-minute walk from Harvard Medical School in Boston Children's Hospital, which is really, really nice because I tend to wake up pretty late in the morning, and this helps me get to work on time. And one last thing that I love about this is they actually have upper year students from Mass Art living here to assist you. So when I walked in, I was having trouble signing in with my Wi-Fi, so they called down an upper year student, and it took about 5 minutes to resolve. So that was really awesome because when you're living alone in the city and in a different country, it's often pretty daunting. So it's nice to know that you have sort of the support system in your residences. And there's tons of other students here from Mass Art and from universities all over the world living here. So it's a great opportunity to meet other students. And yeah, so I'll give you a tour. Alright, so just spend the last 30 minutes on reorganization. I'm really happy with the result. So here's my main study area. I've got a charging port right there, laptop, portrait of some good memories from second year, and these two really comfortable couches. I've got a cabinet with my MCAT notes / textbook. I've got some miscellaneous supplies here, and here is my bedroom. Pretty empty, I know, but you know, I have all the essentials: charging port, alarm clock, my bag, and bed. So yeah, that is the renovated room.
Here I am waiting, I'll have to leave soon. Why am I holding on? We knew this day would come, we knew it all along. How did it?
All right, so after about a 15-minute walk, here we are at the campus of Harvard Medical School. It is huge, it is absolutely breathtaking, and right here is Gordon Hall, probably the most photogenic part of the campus. Surrounding the quad area are different research buildings and different lecture halls. So tomorrow morning I have a meeting with my supervisors at 11. Hopefully I'll be able to convince them to let me film some of the experiments that we do. And yeah, I actually don't have too much for you guys now, but I'll check in with you guys tomorrow morning. And peace. Alright guys, so just finished my first day of work. I'm very tired, but I had a great day today. I met with all the different PhD students and postgrad students at our lab. I met with my postgrad supervisor, who is currently doing a study of fluorescent imaging in mice models of schizophrenia and neurodevelopment. The complement system is a subset of the immune system. So the immune system has innate immunity, which is essentially the immediate or fast-acting response against pathogens, and the adaptive immune system, which is essentially the longer-term response of the body to pathogens, which involves B cells, T cells, etc. And our research is focusing on the complement system, which is essentially a bridge between the innate and the adaptive immune system. So that's pretty exciting. We're doing some fluorescent imaging, and I actually haven't been able to do any experiments yet. I don't have HR authorization to do so. We're just finalizing some paperwork right now. So hopefully I'll be able to start my experiments next week, and I'll definitely keep you guys in the loop for that, but not too many updates on that right now. Another thing is I am very proud of what I made for dinner tonight. I got some Moroccan sweet potato soup and some broccoli, and I got let's open this up. I got a baguette, some meatballs, and some broccoli. So I'm gonna go chow that down later. And for tonight, I am currently working on finalizing a research paper from a previous lab on something called a vaccine certificate. It's essentially a hypothetical solution for the COVID-19 pandemic, where people who are vaccinated might be able to access certain venues earlier than people who aren't vaccinated. So yeah, we're just analyzing the feasibility of this. Is this an appropriate solution for Canada or other countries around the world? And yeah, so we got the paper back from the peer reviewers, and they made a couple comments that we have to respond to now, and hopefully we'll be able to get this paper published within the next month. I also have, let me just check my to-do list. I have to book my vaccine for this summer. I got my first dose of the Pfizer vaccine about a month ago, so hopefully I'll be able to get my second dose in Boston. And I just have to check in with my supervisors with a couple logistic things. So that's it for today, and yeah, so we'll catch you guys tomorrow. Good morning, everyone. So it's currently 10 a.m. I just got back from Boston Children's Hospital, where I got a negative COVID-19 test. My residence actually requires us to get one COVID-19 test per week, so it's really nice to get that over with and to not really have to go out of my way to get it. I'm also going to be getting my second dose of the Pfizer vaccine this afternoon, so I'm really hyped to finally get fully vaccinated and hopefully be able to go on some runs and stuff without needing a face shield or a face mask or anything. Also for dinner tonight, I will be meeting up with some friends. I'm part of a fellowship program here, where it's essentially 20 undergraduate students doing research at different labs at Harvard, and I'm really excited to meet up with some of them for dinner tonight, and I will be heading to work in about an hour, so I will check in with you later. Alright, so it is currently 6:30 on Sunday, June 6th. I really haven't been recording too much for the past couple of days because I got my second dose of the Pfizer vaccine on Thursday, and I got a bit of a headache and some dizziness, so I didn't really have too much energy to record, but I didn't really do too much in the lab anyways this week. I was sort of just shadowing and observing my supervisor, you know, watching her prepare some buffers, watching her prepare tissues and a technique called cryosection, where we essentially take a mouse brain and freeze it in an ice block, and we cut up the ice block into very thin slices, and then we can take those slices and image them using a microscope. So the microscope that we're using this summer is called Mirrorfish. It's essentially a convolution microscope, which in a nutshell produces much more clear images than a normal fluorescent microscope, because it takes images in multiple planes, focal planes. So in essence, it's just a very fancy microscope that we're using. And yeah, like I mentioned before, I didn't really get to do an experiment myself this week, but I'm hoping, fingers crossed, to start doing some of my own experiments next week. And yeah, so that's pretty exciting. And tomorrow morning, I have a couple things going on. So at 8 a.m., insanely early, I never wake up at 8 a.m., but I have my first lecture tomorrow at Harvard. So essentially the program that I'm going with this summer is the Harvard Immunology undergraduate program. So it consists of research work, so that's everything that I'm doing in the lab, as well as some lectures. So we usually have about 3 lectures per week, but since next week is our first week of lectures, we have lectures every single day, and they're all early in the morning at 8 a.m. So gotta sleep early tonight, so I have energy for that lecture. The lectures are going to be about sort of just the intersection between immunology, neuroimmunology, and a couple other different systems in the body, which is pretty cool, because you actually get to, attend these lectures for free. And I know that Harvard tuition is pretty expensive, so it's nice that we don't really have to pay any extra for this extra bit of information. And yeah, it's just going to be a cool experience. And tomorrow morning at 10:30, I have a meeting with the lab. It's going to be over Zoom. So yeah, we'll probably just cover what we're going to be doing for the next week. And yeah, I'll keep you guys updated.
That you breathe, or things that are on your skin that you don't necessarily want to mount a response to, or the giant number of commensal bacteria that are in your gut and other mucosal surfaces. And so how do we think about things that are this immunologic self versus foreign and dangerous is always this at the heart of understanding immunology.
One man's mood can break another man's soul, or am I just cynical for my own good? Am I too scared to say? Get there for sure.
Alright, so it is currently 6:30 p.m. on Tuesday, June 8th. I had a pretty eventful day at work today. I had the opportunity to finally record in the lab, which is pretty exciting. Before today, I was sort of just observing my supervisors doing different experiments. So it felt a little awkward, sort of just hovering over their shoulders and recording them. But today I had the opportunity to do my own experiments, so I got some pretty cool footage out of that. This morning I woke up at about 7:30 a.m. and got some breakfast and attended a lecture on the human immune system. There were about 65 people in the lecture, and it was over Zoom. And it was really interesting because everyone came from different backgrounds, so different areas of the country, as well as sort of internationally, as well as different stages in their career. So like master students, PhD students, undergrad students, and medical students. And then I came into the lab at about 10 a.m. and watched some mice brain dissections. For this study, I'm not really a part of this study, but it was really interesting just learning how they sort of extract the brains. And for this study, they were interested in looking at the association between inflammatory molecules in the germinator, which is a layer of the meninges, and the onset of lupus, which is an autoimmune disease. And yeah, so they just extracted the germinator from the brain and sort of just scratched away different fragments of the skull in order to extract this tissue layer. And then later in the day, I sort of worked on something called rolling circle amplification. So backing up a little bit, I'm working on a project to determine what the association between this gene called C4 so the association between C4 and schizophrenia. So there's 2 different versions of C4, C4A, and C4B. So what's really interesting is you can take specific primers, and these primers are able to sort of join up with each other for C4A, but not with C4B. So when they join up, they sort of make a circle, and we call this a circular padlock ligation. And so that's what can happen with C4A and C4B. Due to certain amino acid substitutions, it isn't able to circularize, so it isn't able to undergo PCR or any amplification. So when we put our solution into a PCR machine, we're able to amplify exclusively C4A. So that's what I did today, and mixed different chemicals, made different buffers, and so made the solution, amplified the solution today, and tomorrow we are going to be running a gel electrophoresis on this. So fingers crossed, hopefully we'll have some good results, and yes.